It's a pleasure to be here and share with you this broad introduction of the field. I'm going to speak broadly and focus on some aspects of imaging because I know this is what this crowd craves. So yeah, as Johanna mentioned, I am an assistant professor at the University of Cyprus in electrical and computer engineering, and my lab is the Nanotechnology Imaging and Detection Laboratory. So today we'll be talking about molecular imaging of cancer with surface-enhanced Raman scattering nanoprobes. Many new words here, I guess. So we'll start slowly. First of all, why do we care about Raman imaging? So this is an image from a review paper that I did not write. It's written by some friends over from Memorial Sloan Kettering Cancer Center, comparing different imaging modalities. And we can see here like the big things, the MRI, ultrasound things are used in the clinic every day. And then we have here at the very end, this cool guy on the surfboard, and he's doing Raman imaging. And the graph here is set up so that it shows the depth of imaging penetration and the sensitivity. So Raman imaging here is portrayed as having really high sensitivity but poor penetration depth in tissue. And yeah, this is because it's a method based on light. We'll see more on that in the rest of the talk. Another way of categorizing Raman imaging and comparing it with other modalities is by looking at its strengths, that are the resolution and its multiplexing capacity. So this figure here is from a review paper that I wrote on multiplexed imaging. And it shows that it compares different imaging modalities based on how many molecular targets they can image simultaneously and at what resolution. And we see here SERS is not the best on any of these, but it does reasonably well in both counts. So it can image multiple targets at a micron size resolution. Additionally, it is compatible with in vivo imaging. So it's a method that we can use both in extracted fixed tissues, but also in living organisms. And I want to say that we typically use the word Raman imaging and SERS imaging interchangeably because the SERS nanoparticles are the contrast agents for Raman imaging. So sometimes we see Raman imaging, SERS imaging. Most of the time, it's the same thing. All right, so why Raman imaging? It is an optical imaging method, and because of this, it has very high resolution. It is diffraction limited. However, because it is an optical imaging technique, it has poor penetration depth in tissue. It is high sensitivity. It has high sensitivity and high multiplexing capacity. However, these are only there when we're using the nanoparticles, the SERS nanoprobes as contrast agents. So the resolution and the penetration depth are properties of the fact that we're using light for imaging, and the high sensitivity and the high multiplexing capacity are because we're using smart contrast agents. So in my talk, we'll look into the Raman effect, have an introduction, and how we can use this for Raman imaging. Then look into the separate phenomenon, which is surface-enhanced Raman scattering, and then how we can make contrast agents for Raman imaging using SERS. And at the end, we'll see a rather specialized application of multiplexed imaging, and then we'll look into potential ways of imaging deeper. So to look at one of the strengths of the methodology and one of the weaknesses. All right, let's start with the Raman effect. So Raman scattering has everything to do with molecular vibrations. We can think of molecules are little springs that vibrate, with specific frequencies that depend on their bonds and their mass. And each of these has a natural preferred frequency. Each vibration has a preferred frequency that we can express in this way. So this is what molecules do in non-zero temperature. They just vibrate in specific ways. Now, another way of making molecules vibrate is by exciting them with an electric field. And the light is just that, it's an electric field. So when a photon is incident on a molecule, it will make this molecule vibrate, but it will drive the oscillation using a frequency that's inherent to the incident light. So this molecule will become polarized, and it will vibrate based on the frequency of the incident light. However, this polarization doesn't happen in a vacuum. This molecule already vibrates. So we can see that the polarizability of the molecule depends on its initial displacement, and the displacement depends on the way that the molecule vibrates naturally. So we can expand the polarization into this expression, where we have here a term that's being driven at the frequency of the incident light, and then this more complex term that we can use our trigonometry from high school and break it up into these two terms that give us the difference of frequency between the natural vibration frequency and the incident light frequency. But why do we care about polarization? Well, we have to go back to our Maxwell equations, remember our photonics class, and see that in the wave equation that dictates the propagation of the electric field, the change in polarization is the source term. So the source term, the polarization, is what creates new light. So these changes of polarization cause new photons to be created. And these are the scattered photons. So once we have an incident photon on our molecule, this whole complicated procedure happens to produce new photons. And these new photons are either at the same frequency as the incident photon, and this is what we call Rayleigh scattering. So this is the case where the photon that's scattered has the same frequency as the incident photon. And then we have these two different cases of Raman scattering. In Stokes scattering, the scattered photon has less energy, a lower frequency. And in anti-Stokes, we have a higher energy, higher frequency. So this is the idea of how we get light scattered from molecules that has something to do with the molecular vibrations. Now, all of this is classical. We can look at it from the quantum approach, which is a little bit cleaner. And we can see that different modes of vibration correspond to different vibrational energy states. When the photon is incident, it excites the whole system at a higher energy state that then relaxes back to one of these vibrational states. This difference in energy is the difference that we want to measure. It's the difference of this vibration. And the case where we have a relaxation to a higher vibrational state, this is Stokes-Raman scattering. And then when we have a photon with a higher energy being scattered, and then this is the anti-Stokes. But here we can see that we have another extra case where the incident photon corresponds to an actual electronic transition of the molecule. And this is what we call resonance Raman, and I guess you'll hear about this later after my talk. Now, this is really cool because of this movement of the electron in a different orbital, what we get is that we have even higher polarization. So this enhances the amount of photons that are produced. Mind you, the difference in energy, the thing that we measure, is the same, but we get more photons with this mechanism than the other. So it's really convenient to match the energy of our laser to the vibration, to the absorbance energies of our molecule, because we get more signal. So when we collect all of these different photons for a second or two, we get enough of them to build a spectrum. So the Raman spectrum shows how many photons have been shifted by how much energy. So the x-axis is how different is the energy, and the y-axis is how many of those photons. So each one of these vibrations corresponds to one of these energy levels, which corresponds to one of these peaks. So by looking at this spectrum, we can practically identify the molecular bonds and the molecule itself. So this is Raman scattering, and this is an analytical technique used in chemistry. We can use this for imaging. So we have a excitation laser. We can choose what frequency, what wavelength our laser emits at. Then we use beam splitters. We illuminate the sample, collect the scattered light, analyze it in a spectrophotometer, and then we get the spectrum. If we make this sample holder a mechanical stage, we can move it and take pixel by pixel, point by point, take an image. And typically what we do is that this is the spectrum. It comes from a single point of our sample. We select one characteristic peak from the Raman spectrum, and we use the intensity of that to make an image. So this is Raman imaging. Now, in this application, this image works because we have something that's very simple. It's a single cell that has internalized this very specific molecule, a nanoparticle that contains PLGA. So it's very easy to map this one specific peak. However, real samples, tissues, they are much more complicated. So we need to do a little bit more advanced methodologies. This is a recent paper. I'm not involved in this. This is just plain Raman imaging using the intrinsic signal from the tissue. And in this study, different areas of cancerous tissues were imaged, and we can see the different spectra here, different indications of the molecules that are present in the different samples, and then something more complicated than mapping the peaks is needed. So here what they did is that they used principal component analysis. They analyzed all the spectra using PCA. It seems that the different samples cluster in different areas in the principal component space. And then they used these scores, the scores on the principal components, turned them into an image and merged it with this bright field image to get this type of image that show a chemical image of the sample. And this is really cool. And it's using the intrinsic Raman vibrations of the sample to make these images that can potentially differentiate cancerous tissue from healthy. However, it doesn't show us whether we have specific proteins. So this is not good in molecular imaging in the way that we think about it when we do PET imaging. It's not specific for molecules. It just shows us the chemical makeup. So this is what we've seen. Raman imaging can map the molecular bonds. We don't need any sample preparation, have a really good resolution, very good for thin samples. However, biological samples are way too similar. We need to do raster scanning that takes a lot of time. We don't have very much signal, so we need more time, and we cannot deal with thick samples. Can we make contrast agents? If we do, we get higher signal, we get molecular imaging, and the answer is, of course we can, but we need to use surface-enhanced Raman scattering. So what is this surface-enhanced Raman scattering? So this is a different phenomenon. It's based on surface plasmon resonance. So to get surface plasmons, we need a metallic surface. And when excited by incident light, the electrons that are on the metallic surface oscillate back and forth all together, and they enhance the polarization of the nanostructure, and they create really intense electric fields. Now, if we match the frequency of the incident light to the frequency that is best absorbed by our nanostructure, then we get surface plasmon resonance. And this means that most of our light is absorbed and turned into a local electric field. If we play a little bit with the geometry of the nanostructures, then we get these areas that we have highly amplified electric field. We call these areas hotspots. And if we put a molecule in this hotspot, then this molecule is going to experience a massively enhanced electric field. And the Raman signal emitted by this molecule is enhanced by a factor of 10 to the 10 or higher. That is a big number. This means we can see single molecules as long as we put them in the right spot. Now, in our lab, we use this effect to make sensors. And the way we do this, we use nanoparticles, we bring them together, we try to capture the analyte molecules in the hotspots. However, there's a different methodology where we can use this effect to make smart imaging tags that we can use as contrast agents. So in this case, we have a plasmonic nanoparticle that's encapsulated in a glass shell and targeted with antibodies. And this is what we're going to use for our medical imaging applications. So how do we make those and why? So first of all, we need them to be resonant in the infrared. The infrared window is this group of wavelengths here that allow slightly higher penetration into tissues than the optical frequencies. In the optical frequencies, we have mostly blood absorbing all the light. And in the infrared, we have this lower absorbance. So if we use an infrared laser to excite our molecules, then we can get a little bit more penetration depth. So to do this, we use a star. The resonances of the plasmons on the star are in the infrared area, as we see here. Also, with the star, we get free hotspots on the tips. So there's enhanced electric field on the tips. So then we take dye molecule that has absorbance in the infrared. So we take an infrared dye, and then we get resonant Raman scattering in addition to the SERS enhancement. So we get a really strong signal. We capture this inside a silica shell, so to isolate it from the biological medium and stabilize the signal. And then on the silica shell, we can stick antibodies so that we get molecular targeting. So this is the philosophy of how you do it. This is what they look like. They look like stars encapsulated in nice spheres. They have a pretty well good characterization, limit of detection in the 10 femtomolar or so. And we can use these nanoparticles even without targeting. They home to cancer. This is a classic paper from 2012, published by the late Dr. Kircher. And here they used the same nanoconstruct as I described for imaging glioblastoma with MRI, photoacoustic, and Raman. And with Raman, you can see that you can use it during the surgery, during the tumor removal, because you can do Raman imaging on the fly. So as opposed to MRI, you cannot do MRI during surgery. Another approach that came a few years after that is this, where the nanostars, because the nanostars were had the right resonances in the infrared, the limit of detection was higher, which allowed imaging of microscopic tumors. So here is one example where they imaged what is it? The pancreatic cancer. But all of this is without targeting molecules. This works based on nanoparticles being internalized by cancer cells without any molecular targeting. But we can use molecular targeting. This is what is shown here, where we have a model of breast cancer metastasis to the lung in the mouse. And the mouse was administered nanoparticles, SERS nanoparticles, targeting tissue factor. Tissue factor is a marker for cancer. And here we see that the bioluminescence comes from the cancer cells. The Raman map in the intraoperative setting and ex vivo matches this profile. Then what we did is that we administered non-targeted nanoparticles. They didn't stick, and we administered targeted nanoparticles in healthy mice, and again, we got very low signals. So we see that molecular targeting works. In a different application, we wanted to, instead of administering the nanoparticles in the blood, to administer them topically, so inside the peritoneum to image ovarian cancer. So what we did, we decided to target the folate receptor. However, we administer them in the peritoneal cavity, we image and we get signal everywhere. These nanoparticles stick. It doesn't matter if we have the folate receptor or not. They stick everywhere in the cancer, the animal with cancer, and in the healthy control. So what can we do? We use two different contrast agents. Same nanoparticles, different reporters, different targeting molecules. So the red one is targeted against the folate receptor. The blue one is non-targeted, just has PEG on it. And then in a single scan, we get a complicated spectrum. We analyze it. We get an image based on the targeted, one image based on the non-targeted. We divide the one by the other. And like magic, we see that where we have higher expression of the folate receptor, we get higher ratio of our two nanoprobes. So in this way, we can use the specificity of SERS, of Raman imaging, to see something that is hard to see otherwise. So this is one strategy, like ratiometry, to get an image that's otherwise hard to get. All right, so we've seen these applications. I'm going to talk a little bit about more specific application related to multiplex imaging. And after that, we'll see how we can increase the depth of imaging. Most of the discussion in the next few slides comes from a paper that we published in 2022. And it has this very long title because we did very many things in this paper. I'll walk you through it. So the whole philosophy is that if we use one type of SERS nanoprobe, nanoparticle, we use a specific dye to get this very specific spectrum, and then we use antibody against the target, so that when we image, we know every time I see the spectrum, I know that my target is present. So this is the whole idea, and we can generalize this and say, okay, if we can do this for one nanoparticle, maybe I can do it for n nanoparticles. And if I have different reporter and a different spectrum for every nanoparticle and a different target, I know every time I see this purple spectrum, I know I have the purple target. Every time I see the red spectrum, I have this red target, and so on. So I can image many things at the same time. This idea was not new. People knew that Raman imaging can do this. In fact, there was this paper from 2009 published by Cristina Zavaleta. And what they did there, they had 10 different nanoparticles, each one with a different Raman reporter. And each one has a different spectrum, obviously. And what they did is that they injected this under the skin of a mouse, imaged the mouse, and lo and behold, there is these different images. So we can image these nanoparticles in living mice, and we can differentiate them. However, for more than 10 years after this paper, there was very limited progress in the field of multiplexed imaging using Raman. There was, of course, a few applications, but they were very rare in between. Recently, we've had much more interest in this. So there's this paper by Professor Zavaletta's group, by Olga Remina, where they developed 26 different Raman flavors, we call them, different spectra. And then they used 5 of them for imaging different markers on cells at the same time. Then there was this other paper where they developed 32 different flavors and used 10 of them to image 10 different markers in biopsy samples from a very limited number of patients, ex vivo, so, but real patient samples. And then there was this other paper in 2021 where 5 different SERS nanoparticles were imaged in vivo in mice. However, there was no targeting involved. So this paper focused more on quantification of the different nanoparticles versus instead of targeting. So we have a lot of interest in this in the last 3 or 4 years, more or less. And this is very exciting for me because this is something clearly that I like. So in our work, we followed more or less the same strategy as these other groups. However, to my knowledge, our paper is the only one that has complete vertical integration. So we did everything from synthesizing the dyes to using machine learning models to analyze the results. We've done the whole pipeline. So we worked together with some very talented chemists, and they generated this new type of Raman reporters based on chalcogenopyrrollium chemistry. So they look like this. They have a very similar structure. However, we can change chemically the species that we put here on the edges, on the pyrrollium rings, and the length of these crosslinkers. So we can tune the absorbances and the resonances and also their Raman spectra. So this is a family of molecules that can work as Raman reporters, and we have a lot of control on their properties. And we use this to generate 20 different flavors of Raman, of SERS nanoparticles. And we added 8 more commercial dyes to have a panel of 28 different nanoprobes. And then we dealt with a problem where we wanted to image this in a way that made sense. And it's very, we figured out that there was no easy way of doing this. We have to come up with an imaging algorithm. So this is what happens when we have here a very simple image with only 4 of these flavors. If we try to image just one frequency, just one wave number, we get this image that doesn't really help us differentiate where everything is, because they all have a peak around this area. But we can use regression to analyze the spectrum and simplify it and generate images based on these 4 reference spectra plus the plastic of the plate. So we analyze this image spectrally to produce these 5 images. 4 of them are interesting. The other one is a byproduct. We can ignore it. And then we can take these images and color code them. But we don't color code them in a random way. We use a methodology similar to the phasor plot that was featured a couple of weeks ago in your seminar series, where we place the references around the color wheel instead of on the phasor space. And then we have the scores calculated from the regression projected into this color space. So for example, something that has high scores on the reference A, it's gonna come up here and it's going to be painted purple. Something that's halfway between purple and cyan is going to come here and be painted blue. So in this way, we can merge all of these 4 images into this one that shows the not only in a quantifiable way, but also the combinations of the different contrast agents. So this is like a new approach to pseudo coloring. And at the same time, each pixel retains the quantitative information from the spectral perspective. So we decided to use this strategy to image the immunotherapy, because this is a complicated medical question. It's a very good treatment for cancer. However, it is complex, and we need to image many targets at once. We generated a panel of 8 different contrast agents for 8 different targets. And these are the spectra. This is how we analyze them and color them. We developed a mouse model with 2 different tumors. Half of them got immunotherapy, half of them were not treated. They received sham treatment. And here are some images. We imaged through the skin in a non-invasive setting, in the intraoperative setting. We did quick scans, and these are the complicated images that we can get. We see that there is a difference in the colors. This one has more yellows and reds. This one has more blues and purples. And then we excise the tumors, we fix them. The nanoparticles still remain in the tumor, so we can it's the same contrast agent. We didn't reapply. We administer in vivo, they go into the tumor, they stay there. After fixation, they still retain their signal. So we can image this with microscopic resolution, and we can see with not very much imagination that these features are recapitulated in both images. So this internal necrotic area is this one that doesn't have any signal, and then we have like blue all around. Anyway, so we can see that it matches. So we can compare our Raman images to the histology images, and not only to the generic H&E histology, but we can compare it to the molecular target. So for example, this is the Raman image that we got for the nanoparticles targeted against CD11B. And this is the histological image against the same target. And we can see the same features. And here, for example, in the immunotherapy control, in the immunotherapy animal, we can see that this feature is this feature, and these fingers are here recapitulate. So we can get the same information from our Raman imaging as with histology, which is the clinical standard. And not only for this, but for 8 channels at the same time. So these are 7 targeted Raman images plus a non-targeted control. So then we can ask ourselves, okay, so what are the differences between the treated and the non-treated animals? And we can plot the spectra. We can take specific areas from the non-treated animal and the treated animal, and these are the spectra from these areas, analyze them using our references, and get expression profiles on every area. However, these areas, they're in rectangles, so they don't recapitulate the morphology of the tumor. So we wanted to find a natural way of segmenting the image. So we did a superpixel-based segmentation using this algorithm called SLIC, Simple Linear Iterative Clustering, where it's based on K nearest neighbors. And the way we do it is that we say, okay, I want to segment this picture into 100 superpixels. So I optimize this expression using an iterative approach. I declare distance, which brings together the physical distance and the spectral distance. So If two pixels are similar and close together, they get clustered together. If they are similar and far apart, they don't. If they're close together but they're different, they don't. So after these 20 iterations, we can turn our image into this image where instead of having pixels, we have superpixels, and it groups similar tumor areas into groups. And each one of these groups retains the information, the spectral information from the constituent pixels. So we start with these images that have, for every pixel, a spectrum of 1,000 wave numbers. Each one of those got analyzed into 8 scores based on our references, and then we reduce this using k-means clustering. I said the wrong thing before. Using k-means clustering, we reduce it into a smaller number of superpixels, again with 8 scores representing the different markers that they express. And each of these superpixels retains a characteristic profile. So we can ask ourselves, what is the difference between the superpixels from the treated animal versus the control? And we can use this as input to a model, machine learning model, where we input for all the superpixels the scores as the data set, and then we try to identify what is representative of each of the categories, the treatment, the non-treatment, and the no tumor, and then use that information to classify the superpixels into categories and see whether the treatment has had an effect or not. So this is what we did in this paper. We did the whole thing from the chemistry to the imaging to the image analysis. It was a really fun paper. Now, to close, I want to address this issue of imaging deeper. We know that it's a real problem, a real limitation of Raman imaging, but where does this limitation come from? Well, light, when light enters the tissue, it gets scattered by the microstructure of the tissue. So the photons don't go back into the objective of the microscope, but they travel and they exit at different areas. So there is this new approach that came, it's not new anymore. There's this alternative approach that came from the groups of Nick Stone and Pavel Matusek over in the UK, where it's called spatially offset Raman spectroscopy, where you illuminate one side and then you collect the light from a different side. And in this way, you can get more light and with some clever mathematics, figure out how deep it's coming from. So this is called spatially offset Raman spectroscopy. And of course, we can combine this with SERS and we get surface enhanced spatially offset Raman spectroscopy. And Faye Nicholson over in Dana-Farber is doing some exciting stuff on this method. And we imaged through the skull of a living mouse for the first report for in vivo T-source. And yeah, so now new research by that group and others is showing how we can image a little bit deeper at better resolution and overall improve the method. All right, so we've seen all of these different things. We saw what is the Raman effect, how we can use it for imaging, the contrast agents that are these nanoparticles that we call SERS nanoprobes, different applications in imaging cancer. One of the coolest things, multiplexed imaging, potentially in vivo. The weakness of the methodology is the poor penetration depth in tissue. However, there are ways of fixing it. And with this, I would like to thank you for your time and take any questions.